Welcome to Real Learning Center. We have seen various aspects of human physiology. We saw how the digestion happens in the human body. We saw exchange of gases. We saw about the circulation, so on and so forth. One characteristic feature of kingdom Animalia and of course kingdom Protista is that they show some kind of movement. In this video session, we are going to discuss about a very interesting character of kingdom Animalia, which happens to be locomotion and movement. At the first glance, locomotion and movement is pretty similar and might look the same. But we should remember that locomotion is a condition where there is a voluntary movement of an individual from one place to another, from point A to point B, something like where you have displacement associated. You can say that would be locomotion. For example, walking, running, climbing, swimming, these are all the examples of locomotion. So after some time, you will not be in the same position. What about movement? Movement brings about locomotion. But remember that all locomotion is a type of movement, but not all movements lead to locomotion. For example, when I'm talking, I can say that there is movement of my larynx, my voice box, my tongue. That's movement, it's not locomotion. When I eat my food, there is peristalsis happening. Now that would be a movement. So you see that though locomotion and movement sound the same, give almost the same meaning, they are slightly different. So we are going to discuss about locomotion and we are also going to discuss about the movement in today's session. So talking about movement, we can see that within our human body, we have three different types of movements. They are the first one, which is called as the amoeboid movement. The second one, which is called as the ciliary movement, and the third one, which is called as the muscular movement. Now, all three are pretty important and they have to kind of say work in sync for carrying out various functions. Now, do we have any kind of pseudopodia in our cells? Yes, I'm sure you have studied about circulatory system and components of blood, where you would have come across certain terms like macrophages and leukocytes. Now, these are the cells which are seen in our blood. Now, macrophages and leukocytes show amoeboid movement. What does that mean? It means that whenever there is a new antigen, they can kind of modify their structure, maybe also to engulf them and kind of bring about their destruction. So you can see that there is a kind of coordinated movement of macrophages and leukocytes, right? Do not think that ciliary movements does not exist in our body. In fact, all the cells that you see in our trachea are having small, tiny ciliary structures. In fact, they help to move the dust particles from the trachea to the outside. Okay, it enters into the nasal cavity, and that is mediated by the ciliary movement. Not only this, the movement of the egg from the in the fallopian tube coming from the ovary towards the uterus is guided by the cilia that is present in the oviduct. So you see that ciliary movement and amoeboid movement, both of them happen to be a very important type of movement that happens within the body. Very important because they help to maintain our immune system, they help with proper movement of maybe air or dust or the egg and the sperms. Now, with all this, let me tell you about the most important type of movement which we are going to study in detail, and that is called as the muscular movement. Now, muscular movement is where we have certain tissues which we call as the muscular tissue attached onto the surface of the strong skeletal system, the bones in our body. Now, this tissue, the muscles that we have, is having a very special contractile property. And because of this contractile property, you can see that they're able to move within the organism, and the whole organism also can bring movements which leads to locomotion. So you can say that muscles are the ones which are characteristics of almost all the animals, which helps them to move, helps to bring the locomotion. Muscles, you should know that these are mesodermal in nature, in the sense that they are coming from the mesoderm, and they exhibit four different characteristics. They exhibit four different characteristics. The first one is what we call as excitability. The first one is what we call as the excitability. Why is excitability important? Now that is telling the muscles to actually kind of contract. And the second property is once they get excited, you can see that they kind of contract, the property which is called as the contractility. It cannot remain contracted forever. It can kind of come out of the contracted state, and that is what we call as extensibility. And then along with this, you can see that it is not that the cells every time get torn apart when they undergo contraction and expansion and all that. So the last point, property which our muscle cells show, is very important, which is called as elasticity. So these muscular tissues which are there are the ones which help in bringing about locomotion, bringing about movement. And how do they do it? That is mainly because of the properties that they have, like the excitability, contractility, extensibility, and the elasticity. Right? So we are not going to discuss about the amoeboid or the ciliary movement, but yes, we are going to discuss about the muscles, types of muscles in detail. Now, once we know about this, let's talk about the different types of muscles. We have different types of muscles. Now, how do I call them as different muscles? How do I classify them? Now, these are pretty much based on their location. These are also pretty much based on the type of activity that they do. Please note, all of them show the same properties, but depending on their region where they are present, their activities slightly change. And so we have different types of muscles. So muscles are of basically three types. The first type is what we call as the skeletal muscles. Skeletal muscles are those muscles which are attached to the skeletal system. Okay. Now, if you look it under the microscope, which you've already seen in the other previous chapters, the structural organization, you have noticed that there are some light and dark bands. So these are skeletal muscles. Because they are banded, we call them as the striated muscles. Now, because these are attached to the skeleton, I will also call them as the voluntary muscles. And these are the ones which helps in locomotion. Hence, I will also call them as the locomotory muscles. Okay. Please note that these muscles are the ones which help in the change in body posture, change the body's function too. Skeletal muscles, because of striations, I call them as striated. But otherwise, I call them voluntary because I can control their movement. I call them as locomotory because they move and they help to maintain, change body posture, body's function. The next kind of muscles, again classified based on their position, on their place, where they are present in the human body, is something that we call as the visceral muscles. Viscera is the word that we use for all the internal organs. Can be esophagus, can be liver, can be stomach, pancreas. So any organ that is there inside the body, in the abdomen and the thorax, we call them as the viscera. So here, the muscles which make up this visceral organs, okay, is what we call as the visceral muscles. Normally, the visceral muscles are only one part of the organ. Okay, we call that as the endothelium and so many other things. But you see that this is the one which helps in the movement of maybe certain food, or maybe certain uh, air passage, or maybe even gametes, or sometimes even the blood cells. So these muscles are something which make up our internal organs, and we call them as the visceral muscles. Please note, by skeletal muscles we call it as striated, but visceral muscles we don't call them as striated. In fact, we call them as non-striated muscles. So once we know about skeletal and the viscerals, let's go to study about the third type of muscles. And these muscles are called as these muscles are called as the cardiac muscles. Please note again here that the word cardia means heart. So the muscles of our heart is cardiac muscles. They are, I would say, an intermediate of skeletal and visceral muscles to look like. So they show some striations, but not as much as skeletal muscles. They are branched, but not too much branched. That kind of thing. Now, these are also involuntary in the sense that I will not tell my heart to start beating, stop beating, and all that. It happens on its own. Uh, how does it work? Now, that we have already seen in the circulatory system, the various regulatory pathways that our cardiac muscles receive, right? So, these are the different types of muscles that we do see in our human body. And so what we are going to do right now is we are going to study about the skeletal muscles in detail. Talking about the skeletal muscles. Now, why are we studying this and left the other two out? Visceral muscles has been studied in part in digestive system and the circulatory system. Cardiac muscles are also studied in the circulatory system, and the entire muscles in detail is studied in the structural organization. However, because the skeletal muscle structure is slightly different and helps in locomotion, we are going to see what are the different changes that you observe in the skeletal muscles, and we are going to discuss about this in detail. Talking about the skeletal muscles, if you look it under the microscope, you take a section of the skeletal muscle and you see it under the microscope, you see that the muscle itself is made up of certain bundles. So this would be one bundle, there would be one more bundle, one more bundle. They are made up of bundles. And each of these bundles are called as fascicles. Okay, each of these bundles together are called as the fascicles. Now, you can see that these fascicles have a outer covering structure, right? You can see that they are all covered in a membrane-like tissue. Now, that membrane-like tissue is a connective tissue. Okay, connective tissue, which is covering all this muscle bundle, is what we call as the fascia. Okay, it is called as fascia. So if I take a section, I can see lot of fascia. In each fascia, if I take it out, I can see small, tiny, muscle fibers inside. Okay. Now inside this, you are able to see this pink colored structures. Now we call these as the muscle fibers or the muscle cell. I hope this part is clear. So we have fascia. Inside the fascia, we have muscle bundles, fascicles. Inside the fascicles, I will have the muscle fibers. You would have studied when we studied about the structure of a cell, about how these muscle fibers are. They are made up of a cytoplasm, and that cytoplasm is what we call as the sarcoplasm. The cytoplasm which is present inside this muscle fiber is what we call as the sarcoplasm, and every cell has a cell membrane, right? Plasma membrane. Now that is what we call as the sarcolemma. So coming back again, we have the muscle in which there are fascicles. Now this fascicle is covered by fascia. Inside every fascicle, you see a lot of muscle fibers, which has a cytoplasm-like structure called as the sarcoplasm. And each muscle fiber has a plasma membrane, which we call as the sarcolemma. Now, there is something which is very beautiful about our muscle. And what is that? That is inside this muscle fibers tissues, right? You can see that there is a very fine, thin blood capillary that will go inside. Why is this so important? Now, this is the one which takes glucose to the cells, right? So, glucose can enter into the muscle fibers by the beautiful, extensive blood capillaries that are seen inside the fascicle. So, this exactly is how our system of muscles look like. Once we know about how the muscles are, how the fibers are arranged, let's go one step ahead and try to see what is there inside every muscle fiber. So, what is happening inside this small, tiny pink color structure that you're seeing? Let us see what each of this is made. Now, inside, you can see some kind of arrangement of proteins. Okay, now each muscle fiber has sarcolemma, right? Then it has sarcoplasm inside. Now, if you look at the sarcoplasm, you can see small, tiny hair-like structures which are there in the cell. Now, because they're very small, I don't want to call them as fibers anymore. But what I call them is we call them as the myofibrils. Myo meaning it is muscle. I call them as fibrils mainly because they are not as thick as these fibers. They're very small, tiny structures which are there inside. You can see them like small hair-like structures. So I call them as fibrils. Okay. Now, this kind of lines are present in our muscle cells, and that is the reason why if I put it under a microscope, I can see some kind of band, some kind of bands, light band, dark band, you know. So that light and dark band is mainly because of the arrangement of the myofibrils inside the muscle fibers. Okay. Now we know these myofibrils are there. What is this made up of? It has to be made up of something, right? Now, if it is any kind of structural component, it would be a protein. So you see that inside the muscle fiber, I have this hair-like structures, and these hair-like structures are made up of proteins. And if I carefully look at that, we see that there are two types of proteins that are there in the myofibrils. The first protein is what we call as actin, and the other one which we call as myosin. Okay, there is actin and then there is myosin. I want you to note that in this picture, if you see, there are few bands which are very, very light, correct? And you can also see that there are some bands which are thicker, right? As soon as you look at it, you can see that there are thicker bands. So, if you look at the myofibrils, you can see that though they are made up of proteins like actin and myosin, their arrangement might be slightly different. They might give you a lighter structure or the lighter bands. Okay, they might give us the light bands Okay, or they might give us the dark bands. Please note, how will I get it whether it is light or dark? That light and dark is just the arrangement of these proteins, the same proteins, the actin and myosin. Okay, now the arrangement of that proteins is giving me all the difference in their structures. Light band is something which consists of actin only. Okay, it is consisting of actin only, and many times I call this light band as I band. Okay, I call this as I band. So if you look at this structure, you can see that there are some small tiny light bands here and there. I call them as the I band. Similarly, if I'm talking about the dark band, so you can see this is darker, this part is darker, this part is darker, right? Now we say that the dark bands is present here and there, everywhere, which we call them as the EA band. Okay, and it is made up of the protein which is called as the myosin. Okay, now you see that the actin and myosin would be structured something like this. You have my left hand which would be made up of actin, and you have this right hand which would be made up of myosin. Assume that they are light and dark. Okay, so now what would happen is the actin and myosin would kind of come together, form a structure which might overlap, and then they would kind of go away. Okay, so now this is what we call as elasticity, where you have two fibers made up of two different proteins. Now these two proteins would come overlap with each other such that their size reduces, brings about contraction, and they can also move away like this, become elastic, becomes extended, and they would get separated. Okay, now this exactly is how my muscle fibers are arranged inside. Okay, the muscle fibers would be made up of fibrils made up of actin and myosin, which would kind of overlap to give me a different structure. Also note, just like how my palm is having all these fingers, I would have the presence of certain Z lines here and there, which would hold the actin's, uh, you know, pro- protein layer. Okay, so you can see that the arrangement of actin and myosin itself is pretty important to bring about the muscle movement. So now once you know the basic structure of how this is, let us study about how this whole actin, myosin, and all of them they bring about motion or movement. Now, just to complicate the structure, you can see that when I say actin, it is not just one protein that is existing. Please note, if it has to keep moving like this, if it is one single protein, its movement would not be so efficient. Correct? It has to be made up of maybe subunits, or maybe it has to be associated with some other proteins so that I can kind of expand it, contract it, and all that. And somehow, if they have to come together like this right hand finger should be interacting with my left hand fingers, right? Now, if that interaction it has to happen, there has to be some kind of affinity between them, some kind of interactions, chemical interactions that should take place, right? So, though when I say actin, please note that I'm not just talking about one particular single protein, but I would be talking about a short-term polymer. Okay, a protein which remains like a polymer for a short period of time. Okay, now then you would be able to understand the concept of how this moves faster. So let's talk about the first part. Let's talk about the structure of actin. Okay, so when we talk about actin, now actin is polymer. Okay, it is made up of a polymer. Now, if it is a polymer, don't you think it should be made up of monomers? Yes. So there is a monomer which is called as G-actin. The monomer is called as the G-actin. Now, what would happen is this kind of G-actins will come together to form a structure, a polymer structure. Okay. Now, this actin is what we call as a F-actin. Please note, both of them are actin only. One is a monomer, whereas the other one is a polymer, F-actin. So, in the case of F-actin, you can see that there are two polymer chains. Okay, both of them made up of G-actins, but they would form a helical structure which is similar to the green colored helix that you are seeing. So the green colored helix is the F-actin, which is very important because that is the protein which is helping in locomotion. And you see that it is made up of G-actin, but not a single layer, but you can see that there are two actin polymers which are kind of helically wounded to each other. So we have the helically wound F-actin proteins. But you do not see only the F-actins present. Along with that, you see another protein which is the thread-like structure that you are seeing here, the red colored or brown colored structure, which we call as the tropomyosin. Okay, it is called as a tropomyosin. Again, give a thought that myosin and tropomyosin are different. Tropomyosin is the protein that you see attached to the actin filament, F-actin. Okay, and somewhere on the tropomyosin here and there at regular intervals, actually, you can see small, tiny globular structures here. Now, those globular structures are called as the troponin. So, if I'm saying there is an actin filament, right? So, I showed you this picture of so many actin filaments here. Now, every actin filament would be made up of a complex like this, which includes a helically wound actin, F-actin, with somewhere here and there tropomyosins and the troponins that can be seen. Now, this is the structure of our actins. Now, how complicated is our myosin? Is it just a protein? Yeah, kind of. But again, you can see that myosin is also not a monomer. Myosin is also not a monomer, but myosin is also a polymer. Okay, myosin is myosin is also a polymer. Here, this myosin is made up of a monomer which is called as meromyosin. Okay, it is called as the meromyosin. Now, meromyosin would look very much like having a globular head. It would look pretty much like this, a globular head, and you can also see that it has a short arm. Okay, it would have a short arm and it would have tails. Okay, now what is very important is that the head region has a site which can bind to it. So this has a ATP binding site, on its head. So this is how my actins and myosins would be made. Let's now discuss about the most interesting aspect of how muscle contraction happens. So let's talk about the mechanism of muscle contraction. It's a very simple step. I mean, I want to make it simpler, but let me tell you, it's not so easy. Now, the whole of muscle contraction is explained by various theories. We have around 2 to 3 theories which explains about how this whole process of muscle contraction happens. The muscle contraction, one of the best theories that we all agree, is what we call as the sliding mechanism or the sliding theory. So we have seen in a lot of experiments and lot of evidences that this theory kind of pretty much explains to the observations that we made. Okay. Now, according to this sliding mechanism, we say that there is a overlapping of the thin filament and the thick filament. So there is a thin filament, there is a thick filament. Now, sometimes you can see that they will overlap. Sometimes you can see that they do not overlap. Now, this simply sliding over one another is what brings about the muscle contraction. Okay, but cannot be explained so simple, right? So let us just see how this whole setup works for us. Okay, now when we talk about the muscle contraction, I'm talking about the skeletal muscles, and therefore it is voluntary, and therefore I should first get the signal from the central nervous system to a neuron. Okay, so first my brain has to send a signal so that there can be contraction, and it will send it through a motor neuron. Now, this motor neuron releases certain chemicals. We call such chemicals as neurotransmitters. So, there is neuron, and to be very specific, let me write down that it would be motor neuron. Now, this motor neuron comes very close to the fascicles, enters, comes to the myofibrils, okay, and somewhere on the muscle fibers and the muscular myofibrils, it is going to release a neurotransmitter. It is going to release a neurotransmitter. The most commonly sent neurotransmitter would be a chemical or a neurotransmitter which we call as acetylcholine. Okay, there is a release of acetylcholine on the sarcolemma, very close to the sarcolemma. As soon as the acetylcholine is released, then there is the muscle fiber which is sensitive to this acetylcholine, and immediately what happens is there is release of calcium ions. There is release of calcium ions in the muscle cell or in the sarcoplasm. Okay, from the sarcoplasmic reticulum or the endoplasmic reticulum of the muscle cell, there is release of calcium. As soon as calcium is released, okay, as soon as calcium is released, what would happen is this allows to go and attach to the myosin. Okay, so there is calcium which goes and binds to the attachment. Just to recollect, myosin is part of our dark bands, also called as the A bands, right? So we see that it binds to the myosin, but I also just now told you that myosin also has a binding site for ATP. So as soon as calcium binds there, okay, there is also binding of ATP, and then what would happen is this myosin would be pulled back. Okay, the myosin would be pulled back. The myosin would be pulled back, and that causes them to slide over, causes This causes the sliding of the actin filaments over the myosin. But obviously, once they start moving, you can see that the calcium comes out, ATP comes out, and that is when the muscle would again relax. It would look something like this. So I would have uh, this would be actin, and let me say this is myosin. So first there would be binding of calcium onto the okay, this is myosin, so there would be binding of calcium here. Immediately there is some structural change. ATP comes and binds. It releases energy. It binds to the troponin. Then it goes away, comes back. Again there is binding of calcium. It would slide further. It would slide further. It would slide further, and goes to the maximum contraction. Okay. And when there is no calcium and no ATP, then they would again come back. For example, uh, so we have a myosin filament, and then we have a actin filament. In our myosin, we have a head which has ATP binding site, and we also have the calcium binding site. When calcium and ATP bind together, you can see that it attaches to the actin at some place. Okay. And it strongly binds to the actin, forms a very strong bond here. But this cross bridge cannot stay forever. What would happen is, in the process, my actin myosin's head would bend forward, pull the actin slide towards one end. By the time the ATP undergoes, dephosphorylation, energy comes out, its structure again falls back, and it pulls the actin further. So you can see that the helical node is here in the same place, but by the time it has come back here, you can see that it has slided from its position. So for every small moments, you can see that this cross bridges have to be formed, ATP has to be used up, and then it should help in the sliding. So this is exactly how the muscle contraction takes place. I hope this part is clear. Now, I'm sure you would be astonished to find out how much of ATP molecules maybe we are using for even small simple movements. And what we are discussing is only with respect to the striated muscles. There are also non-striated visceral muscles are there, heart muscles are there, and so many others. And therefore, the way our body works is pretty complicated. Just in skeletal muscles, you can see that getting ATP is not the same. For example, in some fibers of our muscles, you can see that there is lot of hemoglobin, which we call as myoglobin. It has lot of mitochondria and produces pretty good amount of ATPs in a very short period of time. Okay, whereas the muscles which are attached to the skeleton, in that region we do not have so much of myoglobin. We call them as white fibers actually. They also have hemoglobin like molecules. They have myoglobin, but they are slightly lesser in number, and they're called as the white fibers. So if you can recollect about what we have learned in the previous classes, maybe you would have realized that when we do lot of muscular activity, there we can get muscle cramps. Which muscles undergo cramps? It is not the red fibers, it's the white fibers. That is because mitochondria is less, oxygen is less, and therefore white fibers are the ones which can very easily undergo some kind of anaerobic activity to give us cramps. Okay, and these white fibers are seen attached to the skeletal system. So this is exactly how our muscles play a very important role in carrying out aerobic respiration, and of course sometimes anaerobic respiration helps in the formation of making a lot of ATP molecules. At the same time, helps us to make and perform various locomotion and movement activities in our body and in our life. But let's not forget that it's not just the muscles. There is also the involvement of the skeletal system. So in our next session, let's discuss about the skeletal system, which is helping for these skeletal muscles to attach and bring about locomotion. Okay, so let's talk about the skeletal muscles next. See you all there. Thank you.